Hello, everybody. It's Mr. Caudill. I hope you're doing well. I'm doing okay. And one reason why is because this is it. It's my opinion that this is the tougher of the two semesters, the one that is all about cell biology in this class. We're done after today. This is it. And you can see behind me on the board, there's not that much. We'll touch on 18.4 and then we will do a little bit of 18.5. As far as chapters 19 and 20 go, it's biotechnology, viruses is 19, which we're not going to cover. I'll let you know a little bit about viruses individually without you reading the textbook. And then biotechnology and then genomes and their evolution are chapters 20 and 21. And we will, again, do that stuff in the classroom. I think that you've read a lot in this textbook. You can refer to the textbook, but the problem with chapters 20 and 21, at least, is that, as I said before, whatever's in here is obsolete. It doesn't matter anymore. Certainly there's some base level stuff, which is fine, but this book is probably a few years old, and there's probably all new discoveries in those fields. So I will catch you up on the latest stuff in the class. We'll just look at journal articles and newspaper articles and stuff like that. Today, okay, so let me let you see what 18.4 looks like. It's just basically about how cells know what to become and the messages that will then cause them to become different tissues versus other tissues. We'll touch on that for a few minutes here. And then we get into a really somber topic, which is really important, and that's cancer. We touched on this in chapter 12 because it's all about cell division or uncontrolled cell division, or as the latest science believes, it's an inability of the cells to die. Cancer, let me talk about the book first so I can put it down. So there's some information here, and I want you to go through this, and I want you to go through it slowly, and I want you to read every word. It touches every one of our lives in some way, and it's hard, and it's not fair, and but I do believe always that knowledge is power. It's important to know what's going on. I want you to read all of this. So anyway, take your time with that stuff, please. Your risk of getting cancer in your lifetime as a male in North America is 50%. As a female in North America, it's 33%, 1 in 3. Here in Colorado, we have a very fit state as far as cardiovascularly, and a lot of people do a lot of exercise, and so they're able to ward off a lot of potential cancers by just being very fit and energetic in their daily lives. But we do have the highest incidence of a certain type of cancer of any other state in America. Does anyone know what that is? You got it, skin cancer. We also have the highest levels of macular degeneration and cataracts, and all of this, I think, is related to UV radiation from the sun and people not protecting themselves. So if you're out doing a sport or something and you can't wear sunglasses or a hat, then don't. I get it. But if you're laying by a pool or you're sitting out with your friends and you guys are just hanging out, put a hat on, put some sunglasses on, and please always wear sunblock. Skin cancer has been a problem with my parents, and I have precancerous things they say on my face, and so I'm very cautious of this. My wife wears sunblock every day of the year, and I was like, why am I going to wear a sunblock in December in Colorado if I'm going to be indoors all day? That's ridiculous. And but I do now. I try to. I think it's just a good habit to get into. Just understand that. Here's what my mom always says: is that her dermatologist tells her that the sins of her teenage years are the things that are haunting her now with her skin and its health. So you may not see any effects of what you're doing now as far as damaging cells, and you may not for decades and decades and decades. But anyway, I guess what I'm trying to let you know here is that there's always a chance to be preemptive and to be proactive and be in charge. Don't let things happen to you. You be in charge of what happens in your life. I guess that's a major theme I'm trying to present here. Well, here's the title of 18.4. It says a program of differential gene expression leads to different cell types in a multicellular organism. And the example they give you in your textbook are these frog eggs. And eventually, how do they know to become a tadpole and then eventually metamorphose into what we know as a frog? But because we don't have a song today, I think the most important thing to begin with, and this happens a lot in people's life, is chocolate. So let me read what I got from Time Magazine. It says, good news for chocolate lovers. Not only can your favorite indulgence improve your overall cholesterol levels, but researchers now know why it works that magic. Japanese investigators have found that polyphenols in coca attach to genes in the liver and intestine, activating the good ones that produce HDL, or good cholesterol, as well as those that help suppress LDL, or bad cholesterol. Not bad. Eat more chocolate. Make sure it's the good stuff, though. So let's talk about how cells know what to be. Specifically, let's start with a movie here, and then I'll show you a couple pictures.
In this video, then, we'll look at the beginning of development, looking into the ovary and an unfertilized egg. It then gets fertilized by sperm, which travel down the fallopian tube. So here are millions of sperm coming along. Several of them will hit the egg and try to penetrate it, but one will win, as it were, go into the nucleus. And then there's a reprogramming process where the male and female nuclei have their genes set aside to be turned on and off for early development. Here you see early cleavage stages occurring, and this is one of the early growth phases. As the embryo moves down the fallopian tube, it's going to form an important stage called the blastocyst here in a few seconds. Of course, in real life, that takes days, about 5 days. At this stage, then, I'd like to draw your attention to the inside of the blastocyst, where there are cells called the inner cell mass, which I'll be abbreviating as ICM. Those are the cells that make the entire animal. And the outer cells give rise to the placenta and other supporting tissues. At this stage, the embryo implants into the wall of the uterus. This is when a pregnancy is really initiated. And now we'll see those blue inner cell mass cells form a disc, and then as the cells continue to grow, they change their physical positions, their kind of geographical relationship to one another. And you'll see that represented here as this disc gets transformed into an embryo. Those lines represent sites where cells are migrating in and out. And here's an important stage when the
Okay, I want to point this out to you. Think of you. Look in the mirror or look at the person sitting next to you and go, oh my gosh, how did I or they come to be so complicated and so put together? And this is what you or they looked like at one point, just a flat disk of cells. The reason why I'm stopping here is because you're going to see these terms again. Ecto means outside, meso I think means middle, that's how I remember it, and then endo means inside. And these are 3 germ layers or layers of cells, and you see maybe the 20 or 30 ectodermal cells on the top. Well, ectodermal cells become things like skin, the nervous system. Mesodermal cells become things like the heart, all the vasculature, the blood vessels, and things like that. And then the endoderm becomes the most inner things that we have, and probably the most inner thing that we have is our digestive tract. And you can imagine there's a lot of components: the esophagus, the stomach, the small and large intestine, the colon. So those are just representative things that I memorize when I see these terms, and specifically when I look at these cells and I say, well, what are these cells going to become? And what you can see here is that they're beginning to be differentiated. And then the question becomes, that will be answered in 18.4, how do they become differentiated? If we started with 1 cell, it divided to 2 to 4 to 8 to 16, how do they know? How does this cell right here know that it and all of its descendants are going to become an arm or part of the digestive system? And we'll get to that in just a second. And he's going to touch on it.
The beginning layers of the embryo, the so-called germ layers, are formed, and I'll come back to that in a few minutes. As development proceeds, there's more growth and movement of cells. It'll begin to form a neural tube. Here it turns.
Girls, if you ever think that he's too hot for you, just remember that he once had a tail that was as long as his torso, and he had a rhinoceros head.
And some months later, of course, this would be born as a young baby.
Okay, let's keep going. So here's a picture from your textbook. It's divided in half vertically, and let's just look at the left side here. When we're studying during embryonic development, a fertilized egg gives rise to many different cell types. What I'm trying to highlight here on the left is that The egg itself has, what do they call them? Cytoplasmic determinants. Basically, these are just proteins that are floating around the cytoplasm of the egg. When the sperm comes in, that's fine. The sperm is going to deliver the chromosomes to the nucleus, and now you're going to have a full set of chromosomes, and all of a sudden the zygote is going to be stoked for about 24 hours, and then for about 20 minutes while it divides into two cells, and then we start to try to get a blastocyst and then an embryo and so on. But the point is that in the cytoplasm of the egg, there are these proteins that are floating around. And when all those divisions start to happen and you get 2 and 4 and 8 cells, they just randomly get, I guess, pushed into certain, I don't want to say a corner of a circle, but a certain area of the circle as these divisions are happening. And they're just a chemical message that one day is going to bind onto genes and say, express these proteins. And these proteins might be ones that will create the brain or create the cardiovascular system or create a leg or create the excretory system or whatever it may be. But just understand they're floating in the egg and it's all random. Here's the other side of this picture. And basically what this is saying is that once cells begin to determine what they are and they start to get information going in that direction, they will also begin to create proteins. Let's say that something's going to become the nervous system and every one of its descendants will be part of the nervous system. Well, proteins will be created by genes being expressed, and then they will actually leave the cell and they will stimulate receptors on other cells and start a signal transduction pathway that will turn on genes in those cells. And it's like this big, long cascade. The products of one reaction will become the reactants in the next as far as stimulating or turning something else on. And so they will induce other cells to become certain things. You can see that here. Again, the products of one reaction will become the reactant or the thing that stimulates the next reaction and so on. This is kind of the big picture here. So go ahead and turn to your neighbor and tell them what you know right now about how cells differentiate during development, please. Well, we've seen this picture before in chapter 12. It's representative of the cell cycle, the steps by which cells take in order to divide, essentially, into two cells where there once was one. In 18.5, we're going to hit it again because we're talking about cancer. And your book says cancer results from genetic changes that affect cell cycle control.
This animation will show how cancer develops. Cancer is caused by the cells of the body changing so that they grow in an uncontrolled way. Click the navigation arrows below the animation screen to play, pause, rewind, or fast forward the animation. Your body is made up of cells. Nearly all the cells in the body have a nucleus, which is the control center of the cell. The nucleus contains genes, which control everything about the cell, including how the cell grows and when it will die. Damage to genes is the underlying cause of cancer. The damage may be inherited or caused by other factors, such as cigarette smoke. The cells of your body divide to make more cells, so that your organs and tissues can grow and repair themselves. The nucleus controls this division. In cancer, the cells divide in an uncontrolled way, and often more quickly than normal cells. In many cases, the cells form a lump called a tumor. Cancerous tumors can spread into surrounding normal tissue. Not all tumors are cancerous. Some do not infiltrate the surrounding tissues, nor do they spread to other parts of the body. These are known as benign tumors. Tumors that invade the surrounding tissue and spread to other parts of the body are known as malignant tumors or cancer. Cancer cells can invade blood and lymph vessels. These provide a route for spread to other parts of the body to form secondary cancers. This is called metastasis.
Okay. Viruses account, or viral infections were where they think about what a virus does. It injects its DNA into a host cell. That DNA then gets incorporated into the host cell's genome. And most often, what the virus wants to do is they want to have the cell, they do this subconsciously, but what happens is that the cell then will recreate the virus and then make thousands of copies of it. And then eventually it will literally blow open and all the viruses will go out. And in an exponential fashion, viruses will go and they will infect more cells. But what happens sometimes is that the viral DNA goes into, by accident, gets incorporated into the cell where there are genes that control the cell cycle, and it might screw that up somehow. And so 15% of all cancers are caused by viral infection. So my point is that's one thing that you can try to ward off if you can. You don't have to be a germaphobe, but just try to wash your hands, get vaccinated every year for a flu shot. That's my opinion. Just give your body and all your vaccines for school-age children. That's my opinion. My point is give your body all of these defenses that you can give it so that if you are exposed to a virus, that it is less likely to give you any trouble. Likewise, smoking or UV radiation, these things can cause damage to DNA. And if the DNA is in a certain area which controls the cell cycle, similar to these genes that we're about to talk about, then you might have a problem. These first genes are proto-oncogenes. And the example proto-oncogene that we're going to write down here is something called RAS. It's just the name of a protein that's created by this gene. What these genes do is they code for proteins which stimulate normal cell growth and division. You got to divide your cells for repair, for growth, for other things. And so cell division is perfectly fine as long as it's under control. When this goes bad, it accounts for the number over here, 30% of all cancers. And I'll show you in a moment how it can go bad. Conversions of a proto-oncogene to an oncogene, and remember the prefix onco means cancer, can lead to abnormal stimulation of the cell cycle. The problem is you don't want the cell cycle to just go and go and go and not have all these checks and balances or be able to stop. Well, how do these things turn to oncogenes? Well, they can be converted to oncogenes by translocation or transposition. What this means is, as we learned in the last chapter, how certain pieces of chromosomes can break off and be translocated onto other pieces. Well, if one of these genes is broken off and then it goes right next to a promoter, and maybe that promoter is for some other gene that does something totally benign in the body, maybe it will stimulate, as a result of all the transcription factors around, maybe it will stimulate this gene to create more RAS that says, hey, divide, 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 than it used to. We can have gene amplification. We saw this also. You can have duplications of genes, and maybe all of a sudden you get a slippage in the DNA when the copying is being done, and all of a sudden you have multiple copies of this proto-oncogene, and so now you have more message that keeps saying divide, divide. And then over here you can have mutations that might cause it to then say, well, keep dividing cells.
Point mutation, the change in a single nucleotide, is just one way proto-oncogenes can become oncogenes. Some cells become cancerous because proto-oncogenes are amplified, a process that greatly multiplies the number of growth-promoting genes, pushing the cell to uncontrolled growth. Other cells become cancerous because the proto-oncogenes are rearranged in a process known as chromosomal translocation. In this process, a gene from one chromosome is stuck together with a promoter region from another chromosome. The resulting fused gene can send a constant stream of growth signals.
Okay, go ahead and tell your neighbor what you know about proto-oncogenes and oncogenes. You know, one of my buddies, Kevin, from Northern California, he had a child before any of us had a child. He was married many years before any of our friend group was. And he had this kid named Cameron. And I used to go over and see Cameron all the time. He was this little guy, and maybe he was 1 or 2 at this time. And I remember him, he would always, he lived on this, my friend Kevin lived on the second floor of a condominium complex. And little baby Cameron would stand on the couch always and look out the window because he wanted to see what was going on outside. He was interested. And so he'd always stand up there, he'd bang on the window and stuff. So one day Kevin was home alone, his wife was gone, and he could hear the little kid was cruising around and everything was fine. And then all of a sudden he couldn't hear anything anymore. He's like, well, that's not a good sign for a baby when you can't hear anything. So he said he walked into the living room, can't see his kid, and he looks at the window and the screen of the window was gone. And he's like, oh, that's a problem. So he walks over to the couch, he looks out the screenless window, and he sees the screen laying down on the parking lot below. This is a second-story window. He then's like, oh my gosh, where's my kid? This is a big problem. All of a sudden he hears this crying, and he's like, I don't see any kid, but I can hear some crying. So he runs downstairs, he runs below his window, he looks up, he doesn't see anything, he sees the screen on the ground, and 30 foot away in a bush is his son crying. And what doctors surmise happened was that he fell out of the window and he landed on his face and he bounced 30 feet into a bush. He had a broken occipital lobe right here, this area underneath your eye and your cheek. But my point is could you imagine driving up in your car, you're parking your car, and you see this baby hit the ground and bounce 30 feet into a bush? Now, when babies are born, they have 270 or 275 bones versus the I think it's like 206 bones that adults have. And why that is for a few reasons. To get through the vaginal canal, they're all more cartilaginous and kind of, I know your long bones aren't even fused together. They're kind of in twos and they come back together eventually. It's just to give the baby more pliability there, and probably if any accidents may occur. You know how babies can just fall down and get right back up and it's not that big of a deal. They'll eventually fuse together, these cartilaginous plates, after a while. But anyway, just wanted to give you something to think about. Let's talk about tumor suppressor genes. These things, they're called the guardian angel of the genome. And basically what these guys do is they create a protein called P53. And what this protein does is it basically keeps the genome healthy. It looks for mistakes. It looks for copying errors. It looks for breaks in the genome. It just keeps everything healthy. And these are called tumor suppressor genes for a reason. They prevent uncontrolled cell growth. They also play a role in anchorage dependence. A lot of cells need to be anchored to something in order to divide, and we've evolved to have this so that we don't have this metastasis of cancer cells that you saw in the little video where they can get in the bloodstream and be free floating, go other places in the body, then attach and then start to divide, and you start to get a tumor somewhere else. But tumor suppressor genes help to control that, and also density dependent inhibition. The point is cells don't like to stack on one another. They do, of course, if you're creating a tissue to a point, but they don't like to stack like hundreds of cells on top of each other. And they will just stop dividing. And tumor suppressor genes play a role in all of this, the proteins that they make. So they're really, really important. Mutations that decrease protein products of tumor suppressor genes can contribute to cancer. You can imagine that if you have a mutation here and you don't make this as much, then you're not going to suppress tumors as well as you once did.
P53 molecule, protein, binds to specific sequences adjacent to genes which it controls. There's a P53 molecule binding to its site, its binding site. It recruits an RNA polymerase, not a DNA polymerase, that then makes RNA from these genes that it controls.
Genes whose products function as the equivalent of cellular brakes also exist. As a group, these genes are known as tumor suppressors. Humans have 2 copies of each gene, one inherited from each parent. If a single copy of a tumor suppressor is damaged, the other copy is usually able to stop the cell from behaving abnormal. This would be like losing either the front or rear brakes of a car. The car may be damaged, but would still be able to stop. But if the second set of brakes is damaged as well, the car would not be able to stop, just as the cell would not be able to stop dividing if both copies of the tumor suppressor genes are damaged.
Here's the good news, if there is a silver lining here, is that it takes a long time for cancers to develop. I understand that some young people get cancer. I get that. But the millions and millions of people, if I tell you 50% of men in this country are going to get cancer, maybe I'm wrong, I hope I am, we're talking about tens of millions of people. We don't see that many individuals with cancer who are young. And what that suggests then is that it takes time in order for cancers to occur in people's bodies. And also then and this is the theme I've been trying to tell you, in my opinion, then it gives you a chance to do something about it, preemptively, live a good life. And let me show you what I'm talking about. Multiple mutations are generally needed for full-fledged cancer. I don't know what full-fledged means. Thus, the incidence increases with age. At the DNA level, a cancer cell has at least one active oncogene. So a proto-oncogene has been mutated, and the mutation of several tumor suppressor genes. I read in your textbook that it's about 5 to 6 that you need mutations here to knock out these things that help suppress tumors. So that means that that the kind of DNA damage that you accumulate throughout your lifetime is going to happen, A, just by living, but B, you can try to do your best to minimize that. You know, you may have a gene that is damaged and it runs in your family and it might cause a certain type of cancer, but that's probably only one of these. And so, yes, you have the deck stacked against you or your family or my family, but we all know now that it takes many of these. And so, I don't know, just do the best that you can, and I'm going to do the best that I can.
Normal cell division in the body depends on a precisely regulated set of events that determine when a cell will divide and when it will not divide. Two types of genes called proto-oncogenes and tumor suppressor genes are primarily responsible for this regulation. When mutated, however, proto-oncogenes can become what scientists call oncogenes, genes that stimulate excessive division. This situation is similar to getting a car's accelerator stuck in the downward position. A cell that experiences such mutations tends to divide more frequently than it normally would. In contrast, mutated tumor suppressor genes can become inactive. A cell that experiences a mutation in a tumor suppressor gene loses some of its crucial braking power. Again, the result is a tendency for the cell to divide more frequently than it normally would. For a cancerous tumor to develop, mutations must occur in several of a cell's division-controlling genes. These mutations disturb the balance that normally exists between signals that stimulate cell division and signals that inhibit cell division. The result is uncontrolled division.
Go ahead and turn to your neighbor and tell them now what you know about tumor suppressor genes and how they work. Well, let me give you some hope here. Again, try to eat well. Certainly, I enjoy going to McDonald's. Well, it's a little bit. I used to eat 5 hamburgers at once when I was 18, but the point is it's fine. But everything in balance, 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 balance. And remember, shop the perimeter. Remember, you are what you eat. How else do you think you get the fuel for your body? Garbage in, garbage out, or great things in, great things out. Fuel the machine.
One way to change our genes is to make new ones, as Craig Venter has so eloquently shown. Another is to change our lifestyles. And what we're learning is how powerful and dynamic these changes can be, that you don't have to wait very long to see the benefits. When you eat healthier, manage stress, exercise, and love more, your brain actually gets more blood flow and more oxygen. But more than that, your brain gets measurably bigger. Things that were thought impossible just a few years ago can actually be measured now. This was figured out by Robin Williams a few years before the rest of us. Now, there's some things that you can do to make your brain grow new brain cells. Some of my favorite things like chocolate and tea, blueberries, alcohol in moderation, stress management, and cannabinoids found in marijuana. I'm just a messenger. What were we just talking about? And other things that can make it worse. It can cause you to lose brain cells, the usual suspects like saturated fat and sugar, nicotine, opiates, cocaine, too much alcohol, and chronic stress. Your skin gets more blood flow when you change your lifestyle, so you age less quickly, your skin doesn't wrinkle as much, your heart gets more blood flow. We've shown that you can actually reverse heart disease, that these clogged arteries that you see in the upper left, after only a year become measurably less clogged. And the cardiac PET scan shown in the lower left, the blue means no blood flow, a year later, orange and white is maximal blood flow. We've shown you may be able to stop or reverse the progression of early prostate cancer, like extension breast cancer, simply by making these changes. We found that tumor growth in vitro was inhibited 70% in the group that made these changes, whereas only 9% in the comparison group. These differences were highly significant. Even your sexual organs get more blood flow, so you increase sexual potency. One of the most effective anti-smoking ads was done by the Department of Health Services, showing that nicotine, which constricts your arteries, can cause a heart attack or a stroke, but it also causes impotence. Half of guys who smoke are impotent. How sexy is that? And we're also about to publish a study, the first study showing you can change gene expression in men with prostate cancer. This is what's called a heat map, and the different colors, and along the side on the right are different genes. And we found that over 500 genes were favorably changed. And in fact, turning on the good genes, the disease-preventing genes, turning off the disease-promoting genes. And so these findings, I think, are really very powerful. They're giving many people new hope and new choices. And companies like Navigenics and DNA Direct and 23andMe that are giving you your genetic profiles are getting some people a sense of, gosh, well, what can I do about it? Well, our genes are not our fate. And if we make these changes, they're a predisposition. But if we make bigger changes than we might have made otherwise, we can actually change how our genes are expressed. Thank you.
You know, it's true. You are what you eat. Please do right by yourself. Don't be a victim. Be a victor. We saw this picture the other day. Don't allow these genes to be turned on. Keep your epigenome healthy. I found another picture on the internet. You don't want tissues being grown where they're not supposed to be. Give your neighbor a hug. I'm about to cry. I didn't think we could do it. I'm glad that we did it. I hope you learned a lot. Where we're going now is evolution, which is my favorite personally my favorite portion of all biology. And then we'll get into plants and human physiology and animal physiology and ecology, which is interaction of organisms in their environments. So it's all big. It's outside of the cell from now on. Congratulations. I'm not kidding. Take a moment here. Pat yourself on the back. Punch your friend in the arm. Whatever you have to do. Congratulations.